Imagine a machine so complex, so microscopic, and so essential that without it, life as we know it simply would not exist. It is churning away inside every single one of your cells right now, reading your genetic code and turning it into you. For decades, this machine was a black box. Scientists knew it existed, they knew what it did, but they had absolutely no idea what it looked like or how it actually worked. And the race to uncover its structure became one of the fiercest, most dramatic competitions in the history of modern science. We are talking about the ribosome. Today, we are diving into Gene Machine by Venki Ramakrishnan. This is not just a dry textbook story. This is a personal memoir of an outsider, a physicist turned biologist, who entered a high-stakes race against scientific giants to decipher the secrets of the ribosome. It is a story of ego, failure, accidental discoveries, and ultimately, the Nobel Prize.
It sounds like a thriller. I mean, usually when we talk about biology, we think of slow, methodical lab work. But Ramakrishnan describes this almost like a contact sport. What really struck me in the beginning is that Venki, as he is known, did not even start as a biologist. He was a physicist. He comes to the United States from India at 19, lands in Ohio, and realizes he is bored with physics. That is a huge pivot. To go from theoretical physics, which is all math and abstract concepts, to the messy, wet world of biology. It is like a carpenter deciding to become a brain surgeon halfway through his career. And he admits he was clueless. There is this great anecdote where he tries to use a pipette for the first time and plunges it into a radioactive solution without a tip. It sets the stage perfectly because he enters this field with zero baggage and zero reputation. He is the ultimate underdog.
Exactly. And that outsider perspective is crucial because the problem he eventually tackles, the ribosome, was considered a graveyard for careers. Let us break down why this molecule matters so much. You have DNA, right? Everyone knows DNA. It is the information storage. But information is useless unless you can act on it. DNA is like a library of cookbooks. But a cookbook cannot bake a cake. You need a chef. The ribosome is the chef. It is the machine that reads the DNA instructions and stitches together amino acids to build proteins. And proteins are everything. They are your muscles, your hemoglobin, your antibodies, the enzymes that digest your food. Every living thing, from a bacteria to a blue whale, relies on ribosomes. If the ribosome stops, life stops. That is why so many antibiotics work by jamming the ribosomes of bacteria. But here is the kicker. For decades, nobody could see it clearly. They knew it was there, but it was just a blurry blob.
This is where the physics background comes back into play, right? Because the tool they use to see this tiny machine is X-ray crystallography. And frankly, this part of the book melted my brain a little bit. Can we simplify this? Ramakrishnan explains that you cannot just use a microscope. Light waves are too big. They just wash over atoms like ocean waves washing over a pebble. You need something with a smaller wavelength, like X-rays. But X-rays do not have lenses. You cannot just snap a photo. So they have to grow crystals? Why crystals? It seems so counterintuitive that you would take a biological machine, which is wet and moving, and turn it into a rock to understand how it works. It sounds like turning a racehorse into a statue to see how it runs.
That is a brilliant analogy, Alex. But here is why they do it. A single molecule is too small and the X-ray signal is too weak. It would be like trying to hear a single person whispering in a stadium. But a crystal is millions of molecules stacked in a perfect, repeating pattern. When the X-ray hits them, they all scatter the light in unison. It amplifies the signal. It turns the whisper into a shout. But you are right. It is incredibly difficult. You are trying to coax a massive, complex, floppy object made of hundreds of thousands of atoms to stack up like bricks. Most people thought it was impossible for the ribosome. They laughed at the idea. They said the ribosome is not a rock, it is a machine with moving parts. It is never going to crystallize. And this is where we meet one of the most fascinating characters in the book, Ada Yonath.
Ada Yonath. She really comes across as a force of nature. While all the big shots in Cambridge and Yale were saying it could not be done, she was just doing it. She was laboring away in obscurity. And she does something really clever. She looks for ribosomes and extremophiles, bacteria that live in extreme conditions. Her logic was that if a bacterium lives in the scalding hot springs or the ultra-salty Dead Sea, its internal machinery must be tough. It must be stable. If it is stable, maybe it will pack into a crystal better. It is such a smart, practical leap. But even with her crystals, for years, she was getting fuzzy pictures. Ramakrishnan describes going to these conferences where she would show these low-resolution blobs, and people would just roll their eyes. She was the woman with the impossible dream, and for a long time, she was alone in the wilderness.
And she faced another huge problem. The X-rays destroyed the crystals. You spend months growing this tiny, precious crystal, you put it in the beam, and zap. It fries. The data disappears before you can collect it. It is like trying to read a book while the pages are burning. This is where the technique of cryocrystallography changed everything. They figured out that if you freeze the crystal instantly in liquid nitrogen, or liquid ethane, you can protect it. You stop the atoms from vibrating so much. You stop the radiation damage from spreading. Suddenly, crystals that would die in seconds could survive for hours. This was the game changer. And in 1991, Ada Yonath publishes a paper showing that her crystals, when frozen, diffracted to 3 angstroms. That is the magic number. That is the resolution where you can start to see atomic structures. And that is when the race truly began.
So the starting gun fires. The sharks smell blood in the water. But where is our main character, Venki, during all this? He is on sabbatical. And this part is fascinating because he goes to the mecca of structural biology, the MRC Laboratory of Molecular Biology in Cambridge, England, the LMB. This place is legendary. It is where Watson and Crick figured out DNA. It is where Max Perutz invented protein crystallography. Venki describes the culture there, and it is intense. They do not care about publishing lots of little papers. They ask one question: why are you doing this? If the answer is not to solve a fundamental mystery of life, they are not interested. Venki learns the ropes there. He learns that he needs to stop playing small ball. He needs to swing for the fences. And he learns a specific technique called MAD, multi-wavelength anomalous diffraction.
Right, the MAD method. This gets a bit technical, but think of it this way. When you shoot X-rays at a crystal, you get a pattern of spots. That tells you the intensity of the waves. But it does not tell you the phase. It does not tell you if the wave is at a peak or a trough when it hits. You have lost half the information. It is called the phase problem. It is the curse of crystallography. For decades, people solved it by soaking crystals in heavy metals like gold or mercury, hoping the metal would stick somewhere and give them a reference point. It was called soak and pray, literally. But MAD was different. You use a synchrotron particle accelerator to tune the X-ray wavelength. You target specific atoms, like selenium, inside the protein. You can mathematically deduce the phases without the messy soaking. Venki realized this was the key. If he could use this on the ribosome, he would have a scalpel instead of a sledgehammer.
But he is still a small fish. He goes back to the US, moves to Utah, and starts setting up his lab. Meanwhile, the big conference in 1995 happens in Victoria, Canada. This sounded like a showdown. You have the crystallographers, like Ada, who are still struggling with their maps, and then you have the electron microscope people. They are producing these detailed three-dimensional images of the ribosome, but the crystallographers call it blobology. They're like, that is cute, but it is just a blob. We want atoms. However, there is a smell of failure around the crystallography camp. Ada had the crystals, but her maps were not making sense. Venki describes asking her a pointed question about why she could not see the DNA grooves, and she gets defensive. It felt like the community was losing patience. They had the crystals, but they could not solve the puzzle.
And then came the Seattle meeting in 1996. This was the turning point. Ada Yonath gets up on stage. Everyone expects a breakthrough. She has these amazing crystals. She talks for 20 minutes about a contaminant protein called enolase. It was not even the ribosome. It was a mistake. The audience was stunned. And sitting in that audience were the heavyweights, Peter Moore and Tom Stites from Yale. Tom Stites is a legend. Big, imposing, confident. He had solved massive structures before. He looks at this situation and basically decides, enough is enough. If she cannot do it, we will. He enters the race. And that is terrifying for someone like Venki. Stites has the resources of Yale. He has a massive team. And he is collaborating with Peter Moore, the guy who knows more about the ribosome than anyone. It is like the Death Star just pulled into orbit.
So Venki is in Utah. He is looking at this. He knows Ada, Stites, and Moore are all fighting over the big part of the ribosome, the 50S subunit. He does the math. He cannot beat them. He does not have the manpower or the head start. So he makes a strategic pivot. He decides to go after the smaller part, the 30S subunit. The ribosome has two parts, the large 50S and the small 30S. The 50S is the factory that makes the protein bonds, but the 30S is the brain. It is the part that decodes the genetic message. It is where the logic happens. And nobody had good crystals of it. It was considered harder, more unstable, but Venki figured, if I go for the 30S, at least I am running my own race. I am not just chasing Stites' taillights.
It was a massive gamble. He had just moved his family to Utah. He left the security of a government lab for the uncertainty of a university where he had to fight for grants. And he is assembling this team. It is not a team of world-famous experts. It is a motley crew. He hires Bill Clemens, a grad student who Venki describes as having dreadlocks and being a bit immature at first. He has a postdoc who is working on chromatin. They are scraping by. But Venki has a secret weapon. Remember the MAD method? The one using anomalous scattering? He did a calculation. He realized that if he used certain rare earth metals, lanthanides, they would have a massive anomalous signal, much bigger than selenium. He calculated that he only needed a few of these atoms to bind to the ribosome to solve the phase problem. He called it his magic bullet.
So the stage is set. We are in the late 1990s. The race is fully on. You have the Ada Yonath camp, the pioneers who are stumbling but stubborn. You have the Yale juggernaut with Stites and Moore putting massive resources into the 50S subunit. And you have Venki Ramakrishnan in Utah, the dark horse, betting everything on the 30S subunit and some exotic physics calculations. And we also have Harry Noller, the RNA genius in Santa Cruz, hovering in the background. It is a pressure cooker. And what I love about this book is how honest Venki is about the anxiety. He talks about the fear of being scooped, the fear that you will do 90% of the work and get zero credit because someone else published a week earlier. It is not just about the noble pursuit of truth. It is about survival.
It is absolutely about survival. And recognition. Ramakrishnan writes about the politics of recognition. He mentions the Nobel Prize early on. He knows, they all know, that this is Nobel-worthy work. The ribosome is too fundamental. Whoever sees it first, whoever maps those atoms, is going into the history books. But the Nobel rule is strict. Only 3 people can win. And there are 4 or 5 major players on the board. You can do the math. Someone is going to be left out. This adds a layer of psychological tension to every experiment, every conference presentation. Do you share your data? Do you hide it? Do you collaborate or compete? In the chapters we covered, we see alliances forming and breaking. We see the Russians dropping out. We see the sheer attrition of science.
And that is where we leave off in this section of the book. Venki has just arrived in Utah. He has his strategy, he has his small team, but he is still climbing the mountain. The ribosome is still a mystery. The map is not solved yet. It is a cliffhanger. We know he succeeds because, well, spoiler alert, he wrote the book and won the Nobel Prize. But reading the details, you realize how close he came to failure. How many times the crystals could have just not worked. Or how easily Stites could have pivoted and crushed him. It gives you a profound respect for the precariousness of scientific discovery. It is not a straight line, it is a zigzag through the dark.
Absolutely. Gene Machine is a reminder that science is done by humans. It is driven by curiosity, yes, but also by ambition, jealousy, insecurity, and friendship. Ramakrishnan is remarkably honest about his own flaws and his own doubts. He does not paint himself as a genius who knew it all along. He paints himself as a guy who kept asking questions, kept learning new tricks, and refused to give up when the giants entered the room. Next time you take an antibiotic, or just take a breath, think about the ribosome. Think about the billions of years of evolution that built it, and the few decades of frantic, human effort it took to finally see it. It is a masterpiece of nature, and decoding it was a masterpiece of science.
Well said, Sarah. That wraps up our deep dive into the first half of Venki Ramakrishnan's journey. From a physics dropout in Ohio to a ribosome hunter in Utah, it is a wild ride. And for anyone listening who thinks science is boring, I hope this changed your mind. This is as cutthroat and exciting as any political drama. We will be back next time to talk about the finish line, the structure itself, and the aftermath of the Nobel Prize. Until then, keep asking questions and keep looking closer at the world. You never know what complex machinery is hiding in plain sight.